Today, we're going to be talking about how we can use model building techniques to study operons. An operon is a cluster of genes that is under the control of a single promoter. These genes are then transcribed into a single mRNA molecule. Operons are used only in bacteria to control responses that require multiple genes, but they are not used in eukaryotes. Depending on the class that you're in, you might be exposed to a variety of different operons with different control mechanisms and different responses depending on their functions. Today, I'll be using the lac operon, which helps bacteria digest lactose, as an example. But by the end of this video, you should be able to produce a model for any operon that you encounter. The first thing you'll want to do is draw the operon and all of its components in the correct order. In the case of the lac operon, there is an inducer that will bind to the operator when lactose is not present to prevent transcription of our structural genes. I like to think of the inducer as an elephant blocking the train tracks. Without lactose, RNA polymerase will never be able to reach the structural genes. If lactose is present, however, the inducer will bind to the lactose, leaving RNA polymerase's pathway open for transcription. One important thing to note is that glucose is the bacteria's preferred food source. So even if lactose is present, the cell will always digest glucose first if it is available. That means cyclic AMP won't be available, and our polymerase will keep falling off the train tracks. Now that we know how this operon works, we want to find a quick and easy way to predict its response under certain environmental conditions. And the perfect way to do that is by building yourself a decision-making model. The first thing we need to ask ourselves is whether glucose is present, because it's our cell's preferred energy source. If it's not present, we need to see if lactose is present to understand how it's affecting our operon. Will the operon spend energy to make an enzyme that digests a molecule it doesn't have? Of course not. So if lactose is not present, our digestive enzymes are not transcribed. Now it's your turn. See if you can replicate this study strategy for the tryptophan operon, and feel free to check out more of the Study Lab's videos and resources.